Today's session, as usual, I don't know if you ever attended a CarboHyde Cyclodextrin Masterclass before. This is already the seventh event among those. And today we'll talk about some basic stuff related to cyclodextrin. Surprisingly, we haven't tackled these topics before, so we'll talk a little bit about where cyclodextrins come from, how they are made, and how the chemistry is created. We'll talk much more about their structure and behavior under different circumstances, and then we'll go into analytical and safety considerations. The talk itself will be around 45 minutes, if I can keep the time well, hopefully, and then we'll save some time for questions. If you have any question during the talk, please type it in the chat box. When I'm finished with the slides, I'll go through all of them. Of course, you can also raise them by the end. So, uh, let's start. Uh, as I mentioned, today's agenda will start with the origin of cyclodextrins, how they are made, not the history lesson, but chemistry-wise, then about their structure and behavior, what a cyclodextrin looks like in different environments, how you can analyze them, both the cyclodextrin and the products made with cyclodextrins, and in the end, we'll talk about safety aspects of cyclodextrins. So I'll try to make somewhat interactive the lesson. So I have four questions for you. Would you know what is the key raw material for cyclodextrin production? So creating the basic cyclodextrins, quickly type into the chat box if you know what cyclodextrins are made of. Okay, just waiting a few more seconds. Well, technically, okay, I see starch and sugar. Technically, both can be a source of producing cyclodextrins, but in reality, cyclodextrins are made of starch, corn and potato starch, depending on where they come from. In the US, they use mainly corn, whereas in Europe and Asia, it's mainly potato, but technically any kind of starch would be suitable, or any other sugar, oligo sugar would be suitable, as we'll see a bit later how cyclodextrins are made. So what they look like, very basically, the structure that we know of. Cyclodextrin come in three sizes, depending on the number of sugar units that just I'll try to use. Okay, anyhow, that compose the cyclodextrins, so that can be six, seven, or eight sugars. At least those are the ones that are in mainstream applications. There are cyclodextrins of smaller cavity size, and there are ones which are larger, but these are not commercially available. And there are, at least as of now, there are no very efficient production routes to create those cyclodextrins. So alpha 1,4 aliphatic glycosidic bonds link these sugars together to create this ring or truncated cone-like structure. When there is no chemical modification to the cyclodextrin, of course, this AI-generated picture is not accurate of the chemistry, but don't be very strict about this one. So when there is no chemical modification of the cyclodextrin, we call them native or natural cyclodextrins, and these can be modified chemically, as we'll see. What is the essence of the cyclodextrins, is the cavity of the cyclodextrins, and the basic difference uh, or yeah, the basic difference between the inside of the cavity and the outer surface of the cyclodextrin, because the inside of the cavity has a hydrophobic nature, and that's why it can interact with hydrophobic molecules that well. Whereas the outer surface of the cyclodextrin is very hydrophilic, so it really likes water. It can mingle with water very well. So this kind of genus type feature of the cyclodextrin is what creates the unique applications. And as a result, of course, an active ingredient or any other type of mainly hydrophobic molecule can be placed into the cavity of the cyclodextrin where the interaction is not covalent, so it's van der Waals forces, hydrogen bonding, and in some cases, depending on the cyclodextrin and the guest ionic interactions, which stabilize this complex, not permanently, only temporarily. And as a result of the complexation, we observe changes in the properties of the guest molecule, mainly, which can be reduced taste, reduced smell, reduced irritation or side effects, or increased solubility, stability, and so on. We're not talking about complexation today, just so that you can put this into context. So, how is a native cyclodextrin made? As we explained, it comes from starch and bacteria, in particular industrially, Bacillus macerans is creating the cyclodextrins. Um, with an enzyme called CGTase, cyclodextrin glycosyltransferase, which technically means that there is this uh, starch, this oligosugar, and the enzyme cuts the starch into smaller pieces and then links these pieces together, creating the cyclodextrin. The enzyme itself is not specific, so alpha, beta, and gamma, and even other cyclodextrins are created in the same process. There are different types of these enzymes and different types of bacteria that produce different types of these enzymes. So the ratio among the three resulting products or the ratio of the resulting cyclodextrins can be different. Then, from this mixture of cyclodextrins, there are different kind of techniques uh, to select or how to separate them. There are template-based techniques, there are complexation-based techniques because the alpha, the beta, and the gamma cavity has substantial or tendential uh, affinities towards very different type of molecules. So, but technically, you can create insoluble complexes with one of them, or as I said, use different templates, and this can turn the ratio of the cyclodextrin, the yield, the end product very significantly. So this is basically how cyclodextrins are made. As I said, it doesn't necessarily have to be starch. It's economic to use starch, but you could use other sugars too. Uh, why were there any need to modify these cyclodextrins? So technically, these are natural compounds. Nature creates the native cyclodextrins. These are created in different kind of fermentation processes. When we make beer, when we make sourdough bread and stuff like that, the yogurt, cyclodextrins can be created if the proper enzyme and the proper bacteria are in place with the sugars or the starch. Uh, the modification of the cyclodextrin was necessary because the native cyclodextrins are good and are pretty cheap, but they certainly could use some improvement. The basic idea was that they could use solubility improvement. And if we modify the cyclodextrin, and as you can see, each sugar unit has three positions to modify. We'll see a bigger picture about that. When you apply a modification, particularly introducing hydrophilic groups, the solubility of the cyclodextrin can be improved dramatically. And also, introducing new functions to the cyclodextrins can lead to a better complexation properties because typically when a gas molecule fits into the cyclodextrin cavity, usually it sticks out on one end of the cyclodextrin at least. These gas molecules are often very long for the cyclodextrin cavity. And if you have groups on the rim of the cyclodextrin, you can interact with the outer part of the molecule too, making the complex much more better. And there are, of course, other uses too, but dominantly it was solubility improvement. So as I said, three groups can be modified of the cyclodextrin. This can be done in a stochastic manner, so randomly, which is very common for excipient use cyclodextrins like the hydroxypropyl beta, methylated cyclodextrin, sulfabutyl are all randomly randomly substituted. Whereas this can also be done selectively in a more medicinal chemistry type of way. Just put one function on the cyclodextrin or substitute all the sixes but nothing else and stuff like that. So depending on the use, the chemistry of cyclodextrins is very versatile. And I do not wish to go into detail. It's just for your remark, starting from beta cyclodextrin with an intermediate, you can get halogenated azides, primary, secondary amines, acids, technically thiolated compounds. Any kind of cyclodextrin can be created, and we have a huge experience with such modifications. So that's all about the chemistry and the basics. Let's look at the behavior of the cyclodextrin. So we'll talk about solid phase behavior and liquid phase behavior. So what do you think is the highest solubility? Again, type into the chat uh, of a cyclodextrin. So what is the concentration you can reach with the cyclodextrin in solution? Oh, 600 one kilogram of cyclodextrin in a milliliter. That's seems to be a vetability challenge, but maybe you're not that far from um, reality. It's very much dependent on the type of the cyclodextrin. We'll talk about how soluble each of them are in different media, but basically you can create very high concentrations of solutions. So technically you can turn cyclodextrin into a honey like uh, system. So just add cyclodextrin and it would still dissolve, especially with the hydroxypropyl and the sulfobutyl. Cyclodextrin is also the methylated one. So these one can be used in very high concentration, and at that point, usually the viscosity will be the stopper of the concentration. So, as we said, they come in three sizes, and depending on what you would try to incorporate in the cyclodextrin, it would be extremely important which size you put. As you can see, the diameter of the internal cavity is very different. The alpha is suitable usually for small molecules like some gas compounds. Not necessarily even organic compounds. So I don't know, carbon dioxide can be also captured in alpha cyclodextrin, but also fatty acids and other linear molecules. The beta cavity is the most popular because it's more or less a good fit for an aromatic ring, which is a very common component of a lot of compounds, both within and outside the pharmaceutical industry. And gamma is usually for bigger aromatic or bigger hydrophilic structures. But as we said, the modification of the cyclodextrin can be also considered more or less as an extension of the cavity. So it could be that, I don't know, the modification of a beta cavity would it enlarge it uh, figuratively so that it would also encapsulate what otherwise would fit a gamma cavity much better. So when you buy a cyclodextrin, how would it come? It would come as a solid. It would be actually a white or off-white powder. I don't really like, so when we go to an exhibition to cyclodextrin manufacturers, a lot of them would have these, I don't know, these plugs or what is it, with white powders filled into them, which I don't really see as something spectacular, but this is what cyclodextrin looks like. It's a white or off-white powder. The native cyclodextrins can be crystalline or amorphous. The modified cyclodextrins, at least in the pharmaceutical use, are always amorphous, partially because of the production and also because they are, as we discussed, not a single compound, but a mixture of compounds. Usually they are very fine, free-flowing powders, very easy to tablet or capsule, but of course the solid properties can be fine-tuned during the manufacturing process. They have some hygroscopicity, and that's also true in their complex form. So when they are isolated as a complex, so due to their hydrophilic external surface, cyclodextrins attract water. So you have to keep that in mind when you store samples like that. This can go up to, well, depending on the type, 8, 9, 10, or even up to 12, 14% mass percent in the final product. Uh, Mostly they are easy to compress into tablets, and in some cases they're not actually used as a complex, but as a binder in these formulations. Usually they have fairly low bulk density, so they're fluffy a bit, but that's also depending on the manufacturing technology, and they're not too sensitive to heat or okay, humidity if depending on how you store them. So just any dry, cool place could be good, especially if you prevent moisture. And in these cases, their shelf life can be, I don't know, up to a decade even. Uh, Talking about the solution, okay, this is a complex, but it doesn't really matter. So if you dissolve this white powder into a vial, you should have a colorless solution in the end. Most cyclodextrins that are used, well, technically all cyclodextrins that are used in the pharmaceutical industry are soluble in water, mostly in a pH dependent manner. So even the ones that do not dissolve well in neutral pH, in alkaline pH would dissolve much better. The water solubility basically in neutral pH is like this one. So the alpha and the gamma would be between 10 and 20%. The beta cyclodextrin has the lowest solubility among all. So this is the top. It's because of its structure and the hydrogen bonds. It's 1.4%. And as we said, the solubility of the modified cyclodextrins is technically unlimited. If you would want to dissolve them in other solvents, you would face some problems. The native cyclodextrins and the hydroxypropyls would be soluble in DMSO, as well as the methyl, and the methyl and the hydroxypropyl are also soluble in different alcohols, so ethanol, methanol. Yet for instance, the sulfobutyl beta cyclodextrin would be only soluble in water and technically nothing else that's useful for an application. The only sensitivity that they possess, I mean, from a chemical perspective, would be against acidic conditions. That would also apply in solid and liquid stage. So the degradation here um, is not the breaking of the substituents on the cyclodextrins, but the ring opening of the cyclodextrins, which can be detected because, of course, there's a then a reducing OH group created in the process. But ring opening is technically what's happening that leads to a linear oligosugar. Otherwise, they're resistant, as we said, to heat, oxidation, alkaline conditions, and light as well. Uh, Another thing to keep in mind, they are still sugars. So if you create moderately concentrated solutions of the cyclodextrins and you intend to keep them over time, it's better be stored in a refrigerator. Otherwise, bacteria can easily start to grow in them. A few words about the pharmacokinetic properties. The oral bioavailability of all cyclodextrins is very, very low. The highest one is processed by the methyl beta cyclodextrin, which is 12%. All the others are uh, a single unit below 5% technically. And cyclodextrins can only cross biological membranes via passive diffusion, so there is no active uh, transport participating. If you're talking about bioavailability or with other delivery routes, these are higher. So rectally, it would be between, again, derivative dependent, 5 and 30%, and the pulmonary derivative would be fairly high, 60 to 80%. Talking about what happens with them in the body. Uh, they're not metabolized by enzymes. If taken orally, they are excreted unchanged via the feces. If administered parenterally, they are excreted unchanged via kidneys. We'll talk about this one later. Gamma cyclodextrin is digested in the small intestine. That's the only cyclodextrin that's digestible. All the others are non-digestible. So, how to analyze cyclodextrin? Let's take a look at the clock. Okay, we're still on time. I will not go into too much detail with the analytical properties. I would really just like to give you a glimpse of the myriads of tools that are out there and what kind of characteristics we usually test in cyclodextrins. So, how do we get the methods? Firstly, so five cyclodextrins, as you can see, the native ones, alpha, beta, and gamma, and also two that are popularly used, the hydroxypropyl beta and the sulfobutyl beta, have monographs, both in the European as well as the American pharmacopoeia, so you can have official testing methods from them, and there are some monographs in the Japanese for the native ones. If you were to use other type of cyclodextrins, however, methylated cyclodextrin, hydroxypropyl gamma, quite unlikely, but sulfolipo cyclodextrin or shugammadex, there are no monographs for these yet, no official testing methods, so this can uh, be a problem or something to validate or develop yourself rather. So, what kind of tests do we do? Of course, chromatography and dominantly HPLC. What kinds of tests we can run with HPLC? Well, with proper techniques and proper columns, I will not go into details. We can distinguish between cyclodextrins. So here you can see a separation among alpha, beta, and the gamma cyclodextrin. Or if we have a composite mixture, like the sulfobutyl or the hydroxypropyl and the powerful technique, we can separate the different bands of distribution. So here you can see band with one substituents, two substituents, three, etc., and so on. We can trace impurities in the cyclodextrin. Here you can see the, I don't know, probably the main peak of hydroxypropyl beta cyclodextrin and possibly impurity raw material beta cyclodextrin and so on. So these are all possible. The detection modes with the chromatography techniques are not as straightforward, but there are different types of detectors that prove to be very good and useful. The let's say old techniques, maybe not the nicest word, old techniques use refractive index detector. These were the first methods that were developed. Later on, ELSD based methods were created, and more recently, CAD is the technique that's usually used. And of course, MS can also be used as a detection technique. To see the cyclodextrins. These are also, by the way, the exact same methods that are used when we are talking about analysis of cyclodextrins in biological samples. So these are also the tools that you may have. For they have no UV absorption, so you have to look at other possible detection tools. When you're doing chemistry, usually it's more, or at least thin layer chromatography is very reliable, quick and cheap, easy to use to track reactions. It's usually more handy than HPLC in the lab and easier to develop. And there are very nice methods for that one as well. What else is there? Of course, we can use MS, especially for if we want to do a structural analysis, identify new compounds. Usually, it's either Molotov or ESI. MS, you can also see the decomposition of the product in some cases. You can determine molecular weight and even see the distribution. So what the pattern that we saw with the HPLC is also detectable, in many cases even more reliably with mass spec. NMR is also one of our favorite, at least at CarboHyde, especially for structure identification. So to see It can be useful to determine the degree of substitution, the pattern of the substitution, to analyze the structure, and, well, as we will see later on, to analyze the complex itself. I will not get into the details of different analytical techniques, but NMR is usually one of the first methods to use when you want to understand your cyclodextrin. What else? As we said, cyclodextrin can contain a lot of water and a lot of other solvents too, usually originating from the production process, different organic solvents. So, okay, thermogravimetric analysis is usually used to determine these. If you're dealing with cyclodextrin polymers, dynamic light scattering is the typical method to determine the mass of the polymer, but it can be also used with complexes or for aggregation evaluation. Some cyclodextrins have the tendency to aggregate. Not to a horrible extent, but usually the native cyclodextrins are the ones which have these property. Capillary electrophoresis is also included in some monographs, either to determine the degree of substitution or some impurities, typically, but it's also nice to study the complexes. IR is a very popular and frequently used identification techniques about the reducing sugars. I already mentioned that the degradation of the cyclodextrins can be traced with these methods, although I must also say that there are very good and, in my opinion, even better HPLC methods to distinguish between the opened the open ringed and the intact cyclodextrins. Okay, water determination, DSC are all on the table. So the other big topic is, okay, how do we test the complex that we make? First of all, do we have a complex at all? How can we make sure? Uh, of course, the complexation itself can be shown experimentally very nicely. So here you can see how a negative gas molecule behaves, how it behaves when a complex is not created properly, and how it behaves when the complex is created properly. So you can clearly distinguish among the two, but of course there are quite a few analytical techniques to help you in that. So, in your opinion, if you ever done that, what would be your first analytical technique, or what kind of analytical techniques do you think you can use to determine if you could create a complex or not? UV, NMR, DSC. Okay, we have a lot of different guesses. I really like that. And you're all right. So technically, any technique that can distinguish between the free form of the active ingredient and the complex form of the active ingredient is suitable to prove the complexation. And I'll show you a few examples for that, how it's done. But these methods are all suitable, of course, with some stipulations and not necessarily for all active ingredients, but they're all suitable to distinguish. I'll show a few examples, but just to explain. So NMR, technically, when you record the spectra of the active ingredient naked and in its complex form, the the part of the active ingredient that goes into the cavity of the cyclodextrin suffers a chemical shift in the NMR spectra. Or similarly, in the HPLC, the elution time of the native active ingredient is not the same as the complex active ingredient. Obviously, it interacts differently with the capillary wall. So, or the column. But okay, I was already thinking forward. The same goes with capillary electrophoresis. So the interaction with the equipment are different. Similarly, the fluorescence or the UV spectra of the active ingredient, if goes into complex, will shift. Um, And you can use this one as a proof of the complexation as well as a basis to calculate the extent of the complexation itself, and so on with all the other techniques. Uh, but some methods are even more straightforward. So if you have a crystalline API, the crystallinity of that API will disappear if you form a complex. The complex formed will be an amorphous environment, as well as you will see a complete change in the DSC thermograms, as one of you pointed out, is a very good tool for that. Similarly, you can use all these techniques to assess the stability change of the compound: thermal, chemical, physical stability. There are a few characteristics of the complex that are often assessed during the way. One is the binding strength, how much is the affinity of the cyclodextrin towards the active ingredient. Here, technically, what you do is a titration. So when you increase the amount of cyclodextrin in a system and you keep the amount of active ingredient intact and you record the spectra, that would mean that you force a higher and higher portion of the active ingredient into the cavity of the cyclodextrin, which technically uh, results in a more pronounced change in the attributes that you record. It's like a titration, technically, until you reach a stage when all the active ingredient technically is incorporated into the cavity of the cyclodextrin, and this behavior can be observed with all these techniques. The second aspect is the stoichiometry of the complex. From the practical point of view, not as critical, I would say, but it's often useful. Most of the complexes on a molecular level are one-to-one, but there are other types of associations present. Here I always say, and you should note that the fact that on a molecular level the complex is one-to-one doesn't mean that in the product it would be one-to-one, because as we said, it's an equilibrium, it's a temporary interaction, and in order to have the gas molecule incorporated in a cyclodextrin cavity at all times, so when the gas molecule jumps out of the cyclodextrin cavity, it should find an empty cyclodextrin immediately, and for that you need excess cyclodextrin. So usually the cyclodextrin is in molar excess compared to the active ingredient. The extent is product dependent. Very useful is, however, in many arguments, not just regulatory arguments, but also in product developments and product improvements, is how the active ingredient fits into the cyclodextrin, which side, because the two sides of the cyclodextrins here you can see are not equivalent. One is smaller, the other one is bigger. Also, the groups to interact with on the surface are different. So which direction, to which extent, what part of the molecule goes into the cyclodextrin cavity. Here there are two techniques basically that you can use: two-dimensional NMR or some in silico modeling. Both are cool and powerful, both have limitations. And of course, the performance of the complex should be evaluated. So how the active ingredient is released from the cyclodextrin cavity, how slow it is, how fast it is, um, how it or if it supports the permeation through membranes and so on. But these are technically the identical methods that you would use with any other formulation. So, the first part of the talk would be about safety considerations. So, what do we know about the safety of the cyclodextrins? Last question. In your opinion, what's the highest approved daily dose of a cyclodextrin? Any cyclodextrin. Highest approved. So, uh, okay, 1.5 gram a day. 2 milligram per kg, that would be like 14 grams a day for an adult male. Several grams. So amandine is very close to the truth, actually. And then depending on the cyclodextrin, for one cyclodextrin, you exactly nailed the number because for the hydroxypropyl beta cyclodextrin, it's exactly 14 grams a day. But for sulfobutylether beta cyclodextrin, it's a bit higher. It's 16 grams a day for parenteral applications. But such huge dose of cyclodextrins is not true for all of them. So let's look first at the administration routes. I do not wish to explain which are the most popular or what kind of cyclodextrins we can use, but highlight that there are products for any kind of administration routes approved except for pulmonary delivery. For pulmonary delivery, there will be also products soon, probably on the market. But as of now, it's the only admin route, inhalation, where there is no product. With all the others, there is. But you can also see that many cyclodextrins have these gaps, and that's not a coincidence. So beta cyclodextrin, you can see it's present in all kinds of products except for parenteral because parenteral beta cyclodextrin is toxic. Or um, well, you can see the methylated cyclodextrin. It has nasal and ocular products. Similarly, not only it has a high toxicity parenterally, but as you may remember, it has fairly high 12% oral bioavailability, which means that even if you take orally, the methylated cyclodextrin would absorb to a fairly high extent that could lead to systematic side effects that should be prevented. The two ultimately safe cyclodextrins are these two in the middle. The hydroxypropyl beta and the sulfobutyl beta cyclodextrin are very well tolerated. You can see the large number of parenteral products. With the sulfobutyl oral and ocular, okay, we should update this. So there are now two approved oral products. And ocular is also rather soon to come. So these are very safe cyclodextrins with limited side effects. As for the others, there are some limitations. Basically, we can say that, okay, again, there is minimal absorption from the GI tract, so there is a very low risk to have systemic side effects with cyclodextrins, except for the methyl cyclodextrin if taken orally. Remember that if we inhale them, it would be a completely different question because we wouldn't know how it gets absorbed, if it accumulates, and how it will be eliminated. But orally, we know pretty much of them. And compared to other excipients, the lack of metabolism, degradation in the body are also advantages of the cyclodextrin. Also, the thing that they cannot cross biological membranes means that they cannot really accumulate in the organs or anywhere else. We'll see if it's true all time or not. When we look at the toxicity, so the three native cyclodextrins, alpha, beta, and gamma, are on the GRAS list. They cannot just be used in parenterals, but they can also be used, oh, in pharmaceuticals, sorry, not parenterals. So they cannot only be used in pharmaceuticals, but also fruit products, nutraceuticals, even components of foods. If you eat too much of the cyclodextrin, the side effect that you're facing is a GI distress. So it acts technically similarly to other sugars. So these osmotic properties create these diarrhea, nausea, potentially. The beta cyclodextrin, as I said, is toxic, has systemic toxicity due to hemolysis. The methyl beta cyclodextrin, as I said also, due to its higher bioavailability, has some systemic side effects. And um, if you apply at high doses um, with the topical products where it's approved, nasal sprays, eye drops, and so on, at 5+%, it can cause local irritation. These side effects of the methyl cyclodextrin are typically associated with its property to interact with cell membranes, and the same goes to beta cyclodextrin. Uh, There are clinical studies where the hydroxypropyl beta cyclodextrin is used in very high doses in humans as an active ingredient. So not as a not as an excipient. As an excipient, it's considered safe in very high doses when it's used as an active ingredient. And here we are talking about mm, 50 grams a day or even more, 70 grams a day, stuff like that. Uh, there are reports that it can cause hearing loss, which raised a very substantial very substantial attention recently, and it's still explored what's the mechanism of action of that effect. We'll talk about renal elimination. So I mentioned to you that if cyclodextrins are administered parenterally, they are not metabolized and all the excretion goes through the kidneys, which is not a problem usually, but can be. I have a separate slide about that. And as I mentioned, two cyclodextrins are very safe in very high doses, technically, with any administration routes. Um, just briefly, if you want to learn more about the safety of the cyclodextrins, there is one very good document, a fairly detailed document that's published by the EMA that you can look into. It goes by administration routes and technically details. Okay, it's not the freshest document, but it's still very detailed. Uh, Contains suggested daily doses and everything else. Highlights all the studies that have been done. Um and this is also the basis what can be done. Interestingly, one might think that orally taken cyclodextrins would encapsulate all kinds of uh, vitamins and other nutrition's intrinsic molecules and interfere with them. But as studies have shown, this is not really the case. So they are not really interfering, at least as we know of today, with other drugs. From a carcinogenicity perspective, they are really safe, and no evidence has been found against that. And as I said, this document contains this EMA document. I can suggest to anyone who's interested in the field. It's very detailed and super professional. Unfortunately, it's quite a few years old by now. I'm not going into this one. As I said, cyclodextrins can be components of different food products. Here you can see the daily doses of the three different type of cyclodextrins that you can use in foods. So alpha, you can use 6 grams a day, beta, half a gram, and gamma cyclodextrin up to 10 grams, which is quite a few. Allergy is an interesting aspect or acute toxicity. Cyclodextrins do not really trigger such immune responses, as at least it's not very common. But we know of occasions. So Sugammadex, which is a cyclodextrin used as an active ingredient, during the clinical trials of the development of Sugammadex, it did cause some anaphylaxis reactions, so some hypersensitivity reactions, not in a very huge amount of cases. I think it was, I'm not sure if I recall it correctly. It was somewhere around 0.4% or so. But still, they did occur, and that was one of the reasons that first it was only approved in the EU and not by the FDA. And also, the case was the same in Japan. Other cyclodextrins, which are used as excipients, do not really produce that kind of reaction. But okay, cyclodextrins obviously can um, potentially do that. As I said, local irritations with some high concentrations with the methyl cyclodextrins might occur in ocular or topical skin applications because they may have some damage on the mucosa. Renal considerations. So I said that if administered parenterally, cyclodextrins would be eliminated via the kidneys. If your kidney is healthy, this is not a problem. If your kidney is not healthy, so with renally impaired patients, this may be a problem, because in that case, the so there are two problems. First, the elimination of the cyclodextrins would be slower, so the cyclodextrins would have more time to interact with the kidney tissue, and that can lead to, or that is known to lead to, some temporary damage to the kidney, and they can contain, yeah, they can contain other molecules incorporated that they bring to the kidney, which will be then at higher concentrations in the kidney. So this is something to consider with renally impaired patients. Um, in general, when we are talking about, okay, comparing cyclodextrins to other solubilizers, as far as um, I know, they are considered that they have a fairly well reputation, and a fairly uh, well suggested, we could say. There are quite a few products on the market, especially parenteral products, which are based on the concept to replace surfactants and alcohols with a cyclodextrin. From one perspective, from um, a life cycle management perspective, but also from a safety perspective, as we said. Okay, apart from, if the kidneys are healthy, technically, the cyclodextrins do not really cause side effects, unlike alcohol. They don't get metabolized, unlike polysorbates. So it's a fairly safe alternative to other variations. Of course, they're not perfect. So, as a take-home message, I think it's good to learn a little bit more, or know a little bit more about the cyclodextrin you are using, not only with the perspective, or from the perspective of how it interacts with the active ingredient, but what else can it interact with, how it will behave when you administer it in the body, what it will interact with in there, how will it eliminate, what kind of internal organs can it interact with, what kind of side effects it may cause, how to analyze it, not only in the complex, but the cyclodextrin itself. And yeah, in general, what to expect when you administer it. So it was a fairly basic psychiatric lesson. I hope you didn't mind that today we were not talking about product development, but this is also part of the story. So thank you for your attention, and feel free to add any questions in the chat box. I'm really happy to answer and go through them. So first, I'll just scroll up and see if any has been raised during the talk. So we'll start with those, and then go from there. Despite cyclodextrins have low oral bioavailability, can it still enhance oral bioavailability of low solubility molecules like curcumin? This is an excellent question, also a very complicated one. So, um, cyclodextrins can improve the oral bioavailability of compounds. With curcumin, there is a tremendous number of evidence for that. But here, it will not be like that in all cases. The question is, what is causing the low bioavailability of the compound? Because cyclodextrins can also act as a permeation enhancers. Today we didn't talk about that one. So there is a solubility enhancing effect and the permeation enhancing effect. The solubility enhancing effect with the cyclodextrins is happening always. I'm not saying that always it's a huge effect, but it's happening always with hydrophobic molecules. The permeation enhancing effect is very product and application dependent. And orally, what happens? So in order to have a permeation enhancing effect, you need a high local cyclodextrin concentration on the mucosa, on the application surface, so that it can interact with the membrane and loosen it up. Orally, when you take it, this will not happen, probably, except if you have a formulation that, I don't know, dissolves in certain type of the gut, and there is a very quick dissolution and a huge boost in the cyclodextrin concentration. But if we are talking about a pill that you swallow and disintegrates in the stomach in 2 liters of liquid, the cyclodextrin concentration would be extremely low to act as a permeation enhancer. It will still help solubilize the material, which will lead to a higher, which will lead in a higher, result in a higher bioavailability, but via solubility improvement, not via permeation improvement. Okay, this question is fairly complicated. I tried to answer it in 1 minute. Hopefully it was understandable. What sort of complex efficiency is possible, and what is a typical molar ratio of cyclodextrin to the active to achieve this? So, I guess, Kieron, you're asking about what is the real ratio of the cyclodextrin and active ingredients in the products. This is very dependent on the affinity that we talked about. If the active ingredient likes to be in the cyclodextrin cavity very much, and it doesn't really want to exit the cyclodextrin cavity, you don't need to add a huge excess of cyclodextrin. There are products on the market that have 1 to 1, but if you are talking about very good parenteral products that have 1 to 1.4 molar ratio of the active ingredient and the cyclodextrin. In some cases, when this fitting is not so good, you have to add cyclodextrins in a huge excess, and I'm not trying to I don't know, hide the sad truth. So in some products, the molar, I think the other end of the reality is one active ingredient to 17 molecules of cyclodextrin. So technically 96% of the product is cyclodextrin, and the active ingredient is an impurity. Both exist. It's very case by case dependent, and dependent on the affinity. How can we determine the ratio and the affinity of the molecule to the cyclodextrin? So usually what we do is what is called uh, um, so technically what it's done is that we um, we create a series of cyclodextrin solutions for each different derivative in an increasing concentration level, usually ending at 25% if the cyclodextrin derivative allows. You add excess of the active ingredient, so an amount that the cyclodextrin cannot solubilize. You stir vigorously for a fairly long amount of time, at least 8 hours, ideally 24, and then you determine the active ingredient that the cyclodextrin could dissolve, and then you picture it in a graph, and technically this would tell you how the different cyclodextrins compare to each other with respect to their solubilizing potency. From this kind of solubility isotherms, you can also calculate the affinity, and there are also other tools that can give you a numeric value of the affinity, but at CarboHyde we usually focus on this kind of approach because it's a realistic information. So the fact that how strong an affinity of a molecule towards a cyclodextrin is will not tell you how much it will solubilize. It will not tell you if it can improve the stability, if it can match the taste. Typically, those are done experimentally. So we just check ourselves, make the solutions, mix them with different types and different concentrations of cyclodextrins, and see how they perform. And from these graphs, you can pick the amount of cyclodextrins that's ideal to you. I don't know. Your target is 1 milligram per mil for the active ingredient. So you can see from the graph, okay, I don't know, 18% of the cyclodextrin would be sufficient to achieve that. And well, as simple as that, we could say. Is size the reason there aren't cyclodextrins available for pulmonary administration? Well, no, not really. So, uh, as I said, pulmonary administration will be probably a tricky route and not many cyclodextrins. So obviously, as you could see, there is a high bioavailability via this administration route. So I would imagine that only cyclodextrins that are parenterally safe would be considered here because, okay, which are not safe parenterally would have systemic side effects. That would leave us two cyclodextrins. Hydroxypropyl beta cyclodextrin is already in clinical phase 2 with different active ingredients for inhalation product. And as far as I know, it's close to commercialization. Even this year, it could become for a medical device, also an inhalation product, or it's a nasal spray. Now I'm a bit confused. I don't know. Anyhow, hydroxypropyl beta cyclodextrin would probably meet it to the market. I'm not exactly sure why there were less developments or not stressing it as much. There are no, in these clinical trials, no, I don't know, severe side effects or anything that would have been a project stopper. For the sulfabutyl, there is one other bottleneck apart from safety, is that sulfabutyl is a polyanion, which means that it can cause to create an isosmotic solution with the sulfabutyl cyclodextrin enables a lower dose of the cyclodextrin itself. So product development is a bit more difficult in this case, but safety could be similarly good. What else we have here? A few compliments. I'm really happy that you enjoyed. Quick question. Is it possible to determine the molecular weight of ionic PCD? I don't know what is PCD. Uh, but well, with SLS, I don't really have information about that. If molecular weight determination is a good tool, I certainly wouldn't choose that one to determine molecular weight, but rather MS. But anyhow, you mentioned concerns about hearing toxicity. How much is it a concern? So it's a huge concern. It has been only recorded for the hydroxypropyl beta cyclodextrin. So the story very briefly was that they used this very huge dose of cyclodextrin, again, much higher than the excipient use, 50, 60, 70 grams per administration into a population of rare disease groups. And after several months of treatment, it caused deafness. So the hearing was gone completely. And then it triggered a lot of research. They compared it with other cyclodextrins, and it was found that other cyclodextrins did not cause this hearing loss, only the hydroxypropyl beta cyclodextrin. But the root cause was never unambiguously discovered. So if it's this particular cyclodextrin that's interacting with somehow the ear tissue or the ear cells, or maybe it's a raw material of the hydroxypropyl beta cyclodextrin that's causing, or I don't know, a component, because this is a mixture. It could be just it's just a component, and modification of the cyclodextrin would lead to otherwise. In any case, it wasn't detected with any other cyclodextrins, and it wasn't detected at the normal excipient use of this cyclodextrin ever. So I would say that it's still safe to use the cyclodextrins as excipient in their approved doses. If you were to consider, for whatever reason, to give very high doses of cyclodextrins to patients, this is certainly something to evaluate since, okay, there is evidence that potentially this is a problem. Okay, Carlo helping me out. How would one find a reliable supplier of cyclodextrins, and what sort of price per kilogram would that cost? Kieron. So, well, depending on the type of cyclodextrin um, that you're looking for, there are, okay, this is not, I don't know, commercial talk. So, there are a few, not too many cyclodextrin suppliers globally. It would depend on what kind of cyclodextrin you're looking for. You can find reliable suppliers in North America and Europe for your cyclodextrins, but you already have a link I can see. Um, How sensitive is the hydrolysis of CDs to pH, uh, particularly on the acidic side from fill? So it doesn't really happen instantly. So, I mean, uh, of course, the lower the pH, the more sensitive it is. There are cyclodextrin-based formulations which are created at pH 2 or 3, uh, and the cyclodextrin is still intact in the end. It's certainly suggested that at the end of the formulation process to raise the pH as high as possible because the cyclodextrin can also break during the storage. So I don't know, years of storage, the cyclodextrin can degrade. Uh, I don't really think that there is a rule of thumb here. If your formulation is acidic, I would say below pH 3 is kept during the formulation process for a period longer than 1 or 2 hours, at least during the development, I would say that it's worth checking if the cyclodextrins have been degraded or not. It's a fairly simple test. Can you send the slides, please? So everyone who registered will receive the recording. The slides we usually do not distribute, but okay, if you drop me an email, I'll send it over. So we'll provide the recording to everyone. If you need the slides, drop me an email and uh, I can send it to you. So we're over time. Thank you all for participating and for this stimulating discussion. I really enjoyed it. I hope you did too, and I could provide some new information about cyclodextrins. You all have a good day, good evening. See you next time. Bye bye.